cellular respiration and energy creation. Ooh, I'm back again. Have you ever wondered how cells make energy? No? Well, don't you worry, I'm gonna tell you anyways. Cells need energy, or ATP energy to be specific. I said this in a previous video because if we don't have energy, we die! And fun fact, dying is not a good thing. So how do we get this ATP energy thing, you ask? Well, to put it simply, sugar. So the next time your parents don't allow you to eat sugar, just tell them you need glucose to survive. But remember kids, everything in moderation. Okay, jokes aside, how does cells make energy? Well, with a process called cellular respiration. So how does this cellular respiration thing work? Well, first, let's just casually ignore the existence of prokaryotic cells and focus on eukaryotic cells that do have membrane-bound organelles like the mitochondria. And that makes my life 10 times easier. Anywho, we now assume that we have at least one glucose molecule and some amount of ADP. And since glycolysis is an endergonic reaction, it means that you need some energy to start. So you first take this one glucose molecule and put it through glycolysis, which is basically a fancy way of saying breaking down glucose with enzymes. And for glycolysis to happen, you first need glucose, 2 NAD plus and 2 ATP. Now we have the ingredients ready. Let's stack these ingredients on a bunch of enzymes and let the magic happen. And now we have 4 ATP, 2 pyruvates, and 2 NADH. Okay, now glycolysis is over and we move on to the next step. Now this is where we have fluor-5 spiral abyss diverge. two different ways to continue this process. We will see how we can do it with oxygen first, as it is also more efficient. First, we need to transport the 2 pyruvates we got into the mitochondrial matrix. And how? Using the passive transport protein pyruvate transclosis, of course. I honestly have no idea what. Anyway, after that, the 2 pyruvates oxidize and turn into acetyl-CoA after joining with coenzyme A. And carbon dioxide is released, and 2 NADH is produced. Wait, what is NADH? Well, it's NAD+, but add an electron and you get NADH. So now we have some acetyl-CoA and some NADH. What now? Well, it's the Krebs cycle, a process that needs oxygen. Converts the acetyl-CoA to 2 ATP, 6 NADH, 2 FADH, and the 2 FADH thing is basically NADH but fancier. I think at least. Okay, now we have all these stuff, but that's still not a lot of ATP. For a matter of fact, I think we produce more of these fancy letter combination things than ATP. What now? Well, ladies and gents, get ready for the most painful thing in this video. ETC, which stands for, believe it or not, electron transport chain, and actually not electronic toll collection. What is this electron transport chain thingy? Well, it transports electrons, silly. Anyway, electron transport chain is a series of transport chains in the mitochondria that occurs between the inner mitochondrial membrane and the and the intermembrane space, yeah. We also need oxygen for this process. Unrelated, but do you want to know how to say process that require oxygen in a fancy way? No? Don't you worry, because free will is a lie. It's called aerobic. But I digress. Disclaimer, the following images are not owned by me. I'm using Google's diagrams. Google has the diagrams are animated, and there way too much thing going on. Okay, we'll get them at the end. In the inner mitochondria membrane, there are transport proteins, proton pumps, that takes the NADH and takes the electron from the NADH, making it NAD plus again, and the electron goes to an electron carrier, and then the hydrogen ions within the mitochondrial matrix gets pumped into the intermembrane space, and there the hydrogen ions are trapped because ions and phospholipid bilayers are not very good friends, so they cannot diffuse through the phospholipid bilayer of the inner membrane. And at some point during the electron carrier thingy, the electrons from FADH also gets pumped into the mobile carrier and gets transported to the next proton pump. These electrons are used to generate a proton gradient for it to be pumped into the intermembrane space. By it, I mean the hydrogen ions. After going through 3 proton pumps, the electrons that were used to pump the protons over gets transported back into the matrix, and it fuses with oxygen particles that attracts hydrogen ions to make water. That does not make any sense. Just there's something to do with electrons, oxygen, and water. Anyway, if there are no oxygen on the receiving end, the chain comes to a halt and you get no ADP and you die, which is why you need to breathe. The hydrogen ions, since they naturally go down the concentration gradients, and since the intermembrane space is now filled with hydrogen ions, they naturally want to go back to the matrix. And the only way to do so, though, is through an enzyme channel combined called the ATP synthase. These facilitated transport proteins for hydrogen ions spin when the hydrogen ions pass through them, and then it just manually puts ADP and P together, making it a TP, and this process is called oxidative phosphorylation. I probably butchered that. Wait a minute. What is ADP? Well, ADP is ATP but minus a phosphate, which is why ADP plus P equals ATP. ADP stands for adenosine diphosphate, and di meaning 2, so there's 2 phosphates in this ADP thing. And ATP stands for adenosine triphosphate. Tri meaning 3, so there's 3 phosphates, and P stands for phosphates, and therefore ADP plus P equals ATP. And okay, I think stressed this point enough now. So how many ATP can we make with this process? Well, according to the vocab paper, it's 36, but according to Google, it's 38, and according to YouTube, it's anywhere between 30 and 38. So believe what you want. It's around the 34, 6, 8 range-ish, I think. I don't know. Okay, that's great and all. But what if there is no oxygen? Remember how I mentioned after glycolysis we go into 4-5 diverge? I mean, there's 2 different paths that you can take depending on if there's oxygen or not. Yeah, so if there is no oxygen, this is what happens. 2 things could happen. Either anaerobic respiration, anaerobic being a fancy word for not needing oxygen. And anaerobic respiration is just take something like sulfate and use that to replace oxygen. And then just pretend nothing's happened. The other method is a lot more complicated. It is called fermentation. Then there is 2 types of fermentation. For one, you can do glycolysis as usual, but when you have our 2 pyruvates, you can split them into CO2 and 2, I'm gonna butcher this,
acetaldehyde,
and ethanol, which is basically just alcohol. And thus this process is called alcoholic fermentation. After that, the acetaldehyde thing can act as an electron acceptor and take the electrons from the NADH and continuing the glycolysis cycle. But if you are any good at maths and common sense, you realize that this method pales in comparison with the aerobic method, as it lacks the electron transport chain, which produces the most amount of ATP. The other method of fermentation is lactate acid fermentation, which is used by your muscle cells during intense workout sessions when they might not get enough oxygen from red blood cells. It is very close to fermentation, actually. Except after glycolysis, the 2 pyruvates act as electron transport acceptors and turns into lactate. The NADH could continue the process, and this method is also very inefficient when compared to the aerobic method. Now, we talked so much about the mitochondria, but how does one look like actually? Well, there is the outer membrane, the inner membrane with the ATP synthase and the electron transport chain, the cristae, and the matrix. The outer membrane is just a phospholipid layer that encloses the organelle like a regular cell, but like a mini cell inside a cell. The inner membrane has folds known as cristae and the site of the electron transport chain. The matrix is the fluid inside the inner membrane, much like the cytoplasm of regular cells. Redox reactions. These are electron transfers, red being reduced, which means the gain of electrons, and ox being oxidized, which means the loss of electrons, so redox. Okay. So, do you still remember what substrate mean? Okay, so you do know, or not. I don't know, I can't predict the future. But how about substrate level phosphorylation? What does that mean? Well, apparently it means the formation of ATP by straight up slapping a phosphate onto an ADP from an intermediate substrate in catabolism. What is catabolism? It's a metabolic pathway that breaks down molecules, releasing energy. And lastly, yeast, which is a fungi that you use in baking for your bread to rise. You also use them in your alcohol to make them more alcoholic, and other stuff like that. Okay, that is all. Thank you so much for watching this video. Until next time, bye.